Welcome, everyone. Thank you for joining us to another webinar organized by ONI. Today's topic is going to be super resolution fluorescence microscopy used for studies in nanomaterials. For that, we have a very special guest today, Professor Lorenzo Albertazzi from the Eindhoven University of Technology. He's an associate professor at the Department of Biomedical Engineering under the research group of Molecular Biosensing for Medical Diagnostics. He's also group leader at the Institute of Bioengineering of Catalonia, IBEC, in Barcelona, Spain. So in his career, he's been jumping between chemistry and biophysics with a bachelor and a master in chemistry awarded in 2007. And then he finished his PhD in biophysics in 2011 from Scuola Normale Superiore in Pisa, Italy, with the advisor, Professor Fabio Beltram. In 2012, he joined the Eindhoven University of Technology in the Netherlands as a postdoctoral researcher. And later in 2013, he was awarded a fellowship of the Institute for Complex Molecular Systems at this university. And then finally, in 2014, he became a Beni Fellow from the Dutch Research Council. He moved to Barcelona in 2015 to the Institute of Bioengineering of Catalonia to start the Nanoscopy for Nanomedicine group. In 2017, he received a prestigious ERC starting grant from the European Research Council. And in 2018, he was appointed as associate professor at the TU Eindhoven at the Department of Biomedical Engineering with the research group Molecular Biosensing for Medical Diagnostics. So in his lab, he aims to achieve molecular understanding of synthetic materials in the biological environment using optical microscopy and nanoscopy. So his group plans to examine nanomaterials that have potential as cancer therapies and understand their interactions with disease cells in order to identify the key principles needed to design targeted, super efficient cancer treatments. His research has been proven by co-authoring 113 publications in peer-reviewed journals, being corresponding author in 56 of them. With all that, welcome Professor Albertazzi. For the audience, if you have any questions, please post them on the chat. We will collect them at the end of the talk for answering them. That said, Professor Albertazzi, the virtual floor is yours.
Thank you, Laura, for a nice introduction. I will start sharing my screen. Hopefully it works. Yes. And yeah, thank you for a nice introduction, for the opportunity to give this seminar online today. So today we'll talk about super resolution microscopy, and being this event organized by ONI doesn't come as a surprise. But what I think we do a bit original in the field is that our focus is not classical cell biology, axon, cytoskeleton, or chromatin structure, but rather a more chemical and biochemical point of view. So indeed, we use super resolution microscopy to study nanomaterials. And a big motivation is this, and is the field of nanomedicine. Probably you heard about it for sure, but if you type nanomedicine on Google, this is what you find: these fancy futuristic cartoons where you see these robots get injected in the body and will go and fix the disease. So it sounds clearly a beautiful dream, but more science fiction than real. This changing a lot the perception, especially in the last years, is we all know that nanomedicine find big application in health, and is most famous example now is clearly the COVID vaccine. There is nothing else than a lipidic nanoparticle able to delivering mRNA content and elicit immune response against the coronavirus. So this really is boosting the field. And if you think about it, though, it's still very few application nanomedicine make it to the clinic. And one of the reasons there's a lot more to understand all these nano objects to be able to rationally design them and broaden their application. And then when nanoscopy come in place, these objects are small, are complex, have complex interaction with biological media. So I think microscopy can play a big role in this field, and that's what we're trying to do as a group. So I always say that this field is a little bit like a black box. There's a lot of questions, and we still a bit blind and try a bit trial and error to develop these nanomaterials. So we try to hope as a group is to open a bit this black box and understand what's inside. And we do three main things, I think, in this context. The first is using microfluidic technologies to make things high throughput. Both in the formulation of particles, where we try to build libraries of nanoparticles, because if you want to understand the relation between structure and properties and function, you cannot do it in one particle. You need a library of compounds to be studied. So we combine fluidics and microfabrication to produce chips to make basically a small factory of our nanoparticles. And we think to automate this synthesis, we can either automate in automatic fashion complex molecules that will form our materials, or formulate materials itself, like this nanoparticle you see on the bottom right. Now that we have something to study, the supersolution will come in play. And part of our group is focusing on purely studying in vitro these nanoparticles, so before getting them in contact with the biological environment. And that's far from trivial. These objects are small, are made of multiple components, have important features and functionalities. So super resolution microscopy, in particular single molecule microscopy, can really play a role here. And these are some examples of imaging of variety of nanostructures using super res. So this basically using super resolution as a characterization technique to understand what we have in our vial after the formulation of a new material with potential therapeutic application. And then of course comes the more bio part. And this also can think in two ways. First, we use super resolution microscopy to look at cancer cell. We mostly work in oncology, so cancer cells are our target, but not with an eye of fundamental biology, but really to understand the potential target for a therapy. For example, we study the biomarker distribution along the plasma membrane that could be a target for one of our nanoparticles. And of course, we use a variety of super res technique to study interaction between the nanoparticles and cells. So you see, for example, red particles entering green cells, and we should understand that where do they go, how they interact with the biological machineries, if they're active, when there is drugs. So there's a variety of questions about it. And we think really super resolution microscopy is the right technique to do that. Or at least I will try to convince about that during my talk. So to get everyone on board, I think majority of the audience will be familiar with how these techniques works, but I will spend one slide to recap a bit the principle of this method. So what you see in the left is a TIF microscopy image of the surface of a cell, and you clearly see it's blurred. And that's not because it's a bad microscope. That's a top-notch TIF microscope with best optics available. But if you zoom in to small features of the cell, like these protrusions, they will be too small, and you hit the physical limit of optical microscopy. This is the diffraction limit. So these are nanoscale objects, and they cannot be resolved with the classical optics because they fall below the 200, 300 nanometer limit of resolution, limited by diffraction. And that's, again, it's a physical limit. And there's nothing better we can do about it with the optics, but we can try to bypass it using some smart tricks. And that's exactly what super resolution, in particular single molecule localization microscopy, does. So the trick is not to look at all your fluorescent molecules all at the same time, but one at a time. So what you see on the right is the exact same field of view of the left, but now you see these blinking lights, and every blinking light, I can stop frame, is one individual molecule. So every one of these spots is one molecule that we, with some photophysical tricks, can turn on and off. And the point is, when you find a single one, you can fit a Gaussian profile and find the position of the molecule. So if you think about this spot, the position is in this of the molecule is the center of this spot of light. And this phenomenon, this fitting of the position of the dye, is extremely precise beyond the Abbe diffracted resolution. So if you do it for all the molecules of the sample, you can rebuild an image that has much better resolution due to the high precision of localizing molecules. So the image on the left now, the super res image, is nothing else than summing up hundreds of thousands of millions of coordinates of single molecules. And you can appreciate the enhancement in resolution. Now you can perfectly see this protrusion. By the way, this is a cell infected with virus. There's an influenza budding out of a cell. So this is a nanoscale object that cannot be resolved with classical microscopy. So you achieve resolutions in the range of 10, 20 nanometer, depends on the method. But I want to say, the nature of the image is very special. That's what struck me most, and what's interesting most to me of this technique. It's not only an image that you get nicer resolution, nice picture out. It contains a lot of molecular information. You can count how many molecules you have. You can discuss their distribution. So it's really the key technique to access molecular level information. And being trained as a chemist, it's all about molecules. So it's the technique to look at molecules, I think. We didn't invent it, of course, otherwise we'll have a Nobel Prize. So this has been developed way more than a decade ago and awarded Nobel Prize in 2014. What was our contribution to the field is to bring this technique from cell biology that was in its initial application to the fields of materials. And also in chemistry and materials, a lot of questions can be answered with a technique able to resolve molecular level features. So what you see here is a synthetic material. There are nanofibers made of self-assembly. This was with the group of Bert Meyer that is famous to the synthesis and formulation of these fibers. And you can see they're fiber-like objects, but it's again very difficult to resolve the details. And applying one of the super-res technique called STORM, we can now resolve it with tens of nanometer resolution and see much better the features. And this, I think, is the best of knowledge, the first time super resolution has been applied to this type of materials. So this was our start. We proved that we can image materials with super resolution. Can we extend it to any synthetic object, any man-made material? And that's more difficult than what it seems. To some extent, cells, from the point of view of a chemist, are always the same, in the sense that what you stain is, we look at with the protein. Of course, you can look at different type of proteins, but chemically it's always a protein that you stain, and always with an antibody, with immunostaining. So going from an image of a protein A for an image of protein B, it requires much less adaptation of your protocol. But if you think about materials, there's a huge chemical diversity. And as the key of this technique is the labeling and the photophysics of the dye, that requires quite more efforts to go from one material to the other. So a little bit reinventing the wheel every time, because it's an inorganic particle, is very different from a lipidic, is very different from a polymer particle. Nonetheless, starting from these supramolecular fibers, we managed to expand the palette to polymer, inorganic, lipidic particles. In all of these cases, we successfully achieved super resolution, single molecule sensitivity. We also looked into gene delivery carrier, a bit similar to the COVID vaccine that discussed as a case at the beginning. And then, mostly through collaboration, we start to look a bit of any sort of synthetic object, not our key interest, but it was nice collaboration to study crystals, emulsion, hydrogels. So I think this whole palette shows how versatile super resolution is, and to many materials can be applied. So it's a very valuable tool for chemists to study materials that are formulated. I want, before going to show some cases of application of these methods in our group, I want to show two methods we're trying to push in our group. And it's always combining super resolution with something else. In the first case, I want to discuss spectral super resolution imaging. So you know that the microscopy is very good to tell where things are, so tell the position of a certain molecule or particles. But of course, there's a whole community doing spectroscopy, so resolving spectral information, because it's spectral information is very important to tell how things are. For example, there's many fluorescent dyes that can change color with pH or hydrophobicity or polarity, and this tells some functional information about the system. Well, what we want to do is combine the two, because you get hopefully the best of both. And this is how we do it. So we built a setup that is not commercially available, where pretty much is 90% like a standard super setup with TF excitation, infinity corrected, tube lens, and the camera. But just before the camera, we put a grating, so a diffraction element. And what it does is some of the light will just pass through, like you see in the cartoon on the left. So there's a zero order, so you generate an image as before. But part of the light will be diffracted and will create a diffraction spectra in the right part of the camera. So on the image on the right, you see exactly what we see in our microscope. So you see on the left an image of, in this case, is a single nanoparticle, and you see the position. And for every of the particles, there is a stripe that is nothing else than its emission spectrum. So for every object on the left part of the camera, we can resolve its emission spectrum. So we can do at the same time spectroscopy and microscopy at the same time. And this can be combined with the classical paint or storm techniques. So again, on the left, you see blinking lights. These are single molecules. And for every molecule, we get an emission spectrum. So that's basically a very powerful tool. It's a single molecule resolved spectroscopy. And again, combined with the smart probes, we can learn a lot about our system. For example, here we use a dye that changes color with the polarity. So basically, how hydrophobic is one nanoparticle. And imaging nanoparticle will give a completely different dimension. We can resolve the size of the particle as usual, but also can tell some function information. And very interesting here, you see some particles are green and some particles are blue. Basically, the green one are much more hydrophobic than the blue. There's two populations, and the reason is the blue particles don't contain enough lipid contents to be able to fold in the correct way and have an hydrophobic pocket. So basically, it's you get functional information with single particle resolution, and that is a tool we're using now a lot in our lab. The second technique I want to discuss is how to combine super res with other techniques, completely different. So it's called correlative imaging. And the reason is very clear, I think, if you look at this image. On the top, you see a nanoparticle image with DNA paint, one of the super resolution technique. So you see the single dots of the localization events. We label a specific molecule on the particle and can resolve, for example, how many of these functional molecules are on the surface. However, if you look at the exact same particles in electron microscopy, as you see below, the picture looks completely different. We don't see specific molecules, but you can appreciate much better the morphology of the particle. There's a core and a black shell around this peg. And of course, the resolution of EM is still much better. We're talking about few nanometer or sub-nanometer resolution in the case of electron microscopy. So every technique has advantages and disadvantages. And what we want to do with correlative imaging is to combine the two. So what we did here is to take our objects, our particles, image in super res, then take it out of a glass slide, put inside the electron microscope, image in electron microscopy, find the same area, and then nanometrically align the two images. So you can really have one image on top of each other. And now we can look at the morphology and perfect size of the particle in electron microscopy and put on top the function information that specific molecule is labeled with super res. So we try to get basically more information by combining techniques. And that I think is a cumbersome method, is not easy, but it's a very powerful method for the future to analyze materials or biological objects. And can be done in vitro, but can be done also in cells. Here you see again an electron microscopy image as a map, or this gray background that show all the ultrastructure of the cells, membrane, mitochondria, vesicles, and on top in green, the specific object labeled. So basically, you can track where our object of interest is in the context of the cell. Again, very complicated, but very powerful. So that's, I think, is one of the avenues of imaging for the future. So basically, we built a toolbox of super res technique, and we can also combine with spectroscopy and electron microscopy. So it's now a powerful tool of magnifying lens to look at materials. And I will show now two stories of among many that we try to push in our group of how we use super res to study nanomedicine. And the first is in the context of material heterogeneity. So this is another cartoon I found on the web from Northwestern University, and this shows a little bit how chemists think the materials are perfect, all the same, an army of clones, all the same size, all the same functionalization. Probably been building a cartoon, they copy-paste the same particles all over. But are they really so perfect? And we already know from electron microscopy that they're not. If you look at the nanoparticle population, you will see there's, for example, heterogeneity in size. So there's some small particles and some large particles. And the reason is that is a man-made object. We're not as good as nature to build perfectly defined molecular structures. But there's more, is there more about beyond size? And that's a question we start with, because EM is perfectly good to look at sizes, but there's a lot of other features of particles that cannot be measured and maybe heterogeneous as well. And for us, this work from Natalia, my first PhD actually, was very enlightening. So here we want to study the RNA particles, so basically particles to deliver RNA similar to the COVID vaccine. And these are typically made of two components, the RNA, you see in red, and something cationic, could be a lipid, could be a polymer, that complex it because of the opposite charge and pack it into a nanoparticle to deliver. So we did here, we started with a simple question. Are they homogeneous, this particle, in terms of how much RNA is inside, or how much polymer is inside, or how big they are? So what's the heterogeneity of the sample? And to do that, we labeled in RNA in red, a polymer in green, and we start to do two-color super res. And again, you know the trick, low-res image, we cannot really resolve our particles, or a bit blurred. We do two-color super res, and now we can really see individual spherical objects. These are nanoparticles. They're yellow because they contain both RNA and the polymer. That makes sense. But what was striking and really triggered us to think a lot about this is when you do quantification. And I really want to push the concept that just looking at nice super res images is not enough. You should really extract data. So the image analysis, I think, is as important and develop an imaging workflow today in super res. And this is the type of data we extract. I'll guide you through. It is basically a plot where every dot is one nanoparticle. And on the x-axis and the y-axis, you have the amount of polymer and the amount of RNA. So you will see that the particle scatters all around the space because they all have a different content of the interesting molecules. So what's in the top right part of the graph has a lot of polymer and a lot of RNA. On the bottom is less RNA and less polymer. And you will see also by color the size of the particle going from gray to yellow. And it also showed that basically the smallest object have less RNA and the largest object have more RNA. So this is one single vial with one single formulation, and you see the huge heterogeneity. There's particle with a lot, particle with little, small particles, large particles, and these are all combined. So you can imagine that this is actually what is injected to a patient, and it's really, really varied in its function. So you can imagine that maybe some of these particles are extremely functional and do all the job, all the therapeutic performance, and many others will be doing nothing because of too little content of RNA, because they're too large, because of who knows what, and can even be toxic. So I think it's very important to understand the heterogeneity, function heterogeneity of a material. So we push this in this topic further. And another important heterogeneity we want to study is the external functionality. So it's typical of nanomedicine and nanotechnology to take nanoparticles and functionalize on the outside with some functional moiety. For example, antibodies. Antibodies were allowed to bind selectively to a receptor and target the nanoparticles to a specific compartment. For example, try to recognize cancers versus healthy cells. But we start from a very easy question. If you put antibodies on particles, how many are there and how many are actually effective? And that sounds an obvious question for the field, but it was actually impossible to measure so far. When you put antibodies on particles, you don't see any more their signal. The particle cover any spectroscopical features of the antibody, so it was very difficult to directly measure how many are there. Plus, we have, of course, some of them that are perfectly functional, but some of them are badly oriented, they're only partial or non-functional at all, simply because the white tips of the antibody are the one that should be exposed. If the antibody is a wrong orientation, will not be functional. Or if you have misfolding or crowding. So there's a lot of things can go wrong after you conjugate antibodies to particles. So to study this, we came with the DNA paint approach. So imagine that you have a nanoparticle with one or multiple types of antibodies. Some of them are good, some of them are not good. What we had is a probe that is basically the epitope of the antibody, is the protein that is recognized by that specific antibody. And this is tagged with a DNA sequence. So what will happen when we mix these two things is that the functional antibody, the one that are folded, well-oriented, in right position, will fish a probe. and the ones that are not will not be labeled. So we basically tag only the active antibodies on the surface of a particle. And then we can add a DNA strand is complementary that brings dyes. You can do sequential imaging, so you always use the same color but with different DNA sequences. And this will bind and unbind. So basically, it will bind, give you a single molecule signal, will be counted, and then go away. And one by one, we count all the functional antibodies on the surface, and then we can do some counting. And immediately by eye, these are 3 particles from the same batch. You will see that every nanoparticle looks different. Some are more red, some are more green. The distribution is different. So you just, again, give a direct picture of how heterogeneous is the functionality on the surface of nanoparticles. And if you start to count, well, you see by eye that some particles are brighter than others. Means they're more functional. So indeed, you see a very broad distribution. Some particles have 100 antibodies active, and some particles have 1,000. So it's a really, really mixed population. And again, some will be active, and some will be toxic or, best case scenario, useless. Also, we find that the intraparticle heterogeneity. Some particles are perfectly homogeneous functionality, like on the left, and some others are clumps of antibodies on top of each other, so showing also that the synthesis didn't go as well as we planned. So this was a good first start, and then we thought, can we do better than that? Because now we see only the functional, but we cannot tell how many are functional on the total. So to really get the fraction of antibodies functional, we came with a 2-color strategy, where we basically directly label antibodies with 1 color, and is in orange here, and this will stain every antibody, irrespective if it's folded, well-oriented or not. And then the second color is the functional assay I showed you before. So we can give an epitope with a dye, and only the good one will fish the epitope. So basically what we will have is that the red color will give us the total, and the green color only the function. And you can put a ratio and calculate it. Now, a technical complication here is that you have to do this with storm, so with blinking, photophysically blinking dyes. But 2-color storm is absolutely not trivial. The red dyes, the 647 dyes, blink very well, but the green dyes often don't do as well, and it's very insufficient if you want to do a quantitative estimation. So to use both 2 times a functional dye, we use spectral imaging. So basically we label with 2 red dyes, CF680 and Alexa 647. So these 2 dyes are actually quite similar in spectral, but they differ only 20 nanometer in emission. But if you use the spectral setup I discussed before, so combined microscopy and spectroscopy, we'll be able to discriminate even 2 dyes that are so close, and then count in 2 color in 1 pot the total and the functional. So basically we'll do evaluate the 2 spectra, how high is the spectra of functional and how high is the spectra of the total. And this is how the data looks like. As again, I see many particles, they all look slightly different, and I will zoom in on 2, and you see different amount of green and red in these particles. And if you quantify the spectra, you will see that, for example, nanoparticle 1 is quite a lot of functionality, so all the 2 peaks total and function are almost the same height, while nanoparticle 3, for example, is much less functional. So you see that every nanoparticle is a different ratio. It means have a different ratio of functional antibodies over the total. And we're using these methods to compare different formulation, different chemistry. So basically, this want to be a guide for chemists how to develop the best synthetic strategy to maximize functionality on the nanomaterials. So basically, it's a guide for synthesis. So to end up this first story, there is a lot of heterogeneity in objects made by man. We are not perfect synthetic people. Maybe it's not even too bad. In some cases, having a heterogeneous population could be even better. And cells are not homogeneous either. They have different expression of genes and different expression of proteins. So what I know for sure that we want to understand it. So if you want to understand how heterogeneous and what's the composition of the materials you formulate in terms of size, functionality, composition, surface chemistry, etc. And super I think is the real optimal technique to do it. Second part of my talk, I want to discuss our biological target. So it's a more bio-based project, and we have a lot of interest in imaging receptors using paint. And that's because they're our target for when you want to develop particles, we want to make it selective by recognizing receptors on the surface of the cell. Here we have two approaches, both based on paint. So paint, as a reminder, is a technique where you have a floating probe that can reversibly bind to your object of interest, and every time it binds, you see a single molecule and you count it. So the very common technique in the literature is DNA paint. As I showed before, you tag with DNA your receptor of interest and give the complementary. And this is done generally in fixed cells because it's a very slow technique. And this we do it a lot. We get beautiful pictures of the cell membrane where this, for example, is a cancer cell and breast cancer cell, and we stain EGFR, a common biomarker, and every of these cluster of dots you see is a single EGFR on the membrane. So we can really count them, quantify the distribution, it's homogeneous, distribution if it's homogeneous along the cell. This is a very powerful static map we can obtain. But today I want to talk about paint in live cells, where we basically want to have probes that without any pre-labeling float and has a reversible affinity for receptor of interest. And as we are in live cells, after binding, the receptor will move. So we also learn something about the diffusion. And this is how a live cell paint measurement looks like. So this is a cell. You don't see it because it's on the background and it's not labeled, but you will see popping up. And we add these probes that floats, and then they tend to reversibly stick to a receptor. And if you start to play the movie, you will see the single molecule moves because the receptor, of course, is on live cells and float on the plasma bilayer. So you see appearing of dots, tracking, and disappearing. So there's a lot of information here that we can extract. And now you also see the contour of the cells popping out trajectory by trajectory. So we can learn a lot from this data. We can learn about the number of events. That is basically tells something about how many receptors are there. So you can count the events that appear. We can build a map. So again, we can get an idea of the heterogeneity and distribution of these receptors. Of course, it's not a static perfect map as before because diffusion of the receptor make it a bit more blurred. So you sacrifice a bit of spatial resolution, but you can still rebuild an image. You have the tracks. So you can tell something about the mobility of the receptors, how fast it moves. There's a preferential movements, there's Brownian diffusion, there's interaction that stop it. So this reflects a lot the biochemical behavior of the receptor. So it's a very information-rich type of technique. However, the key is that you need reversible low affinity binders that are specific but reversible. If you have the affinity is too high, the probe will never go away and will block the receptor forever, so you cannot measure many. And if it's too weak, it will be impossible to detect it because the events will be too fast. So this is not trivial to get binders in the right regime. In the last years, we look at several based on aptamers and affimers, but today I will focus on one specific story. There's our glycopaint story because it's our most recent work. Very happy to show it. So the principle here is to engineer a physiological interaction for imaging. And in particular, we decide to start with glycan-lectin interactions. So, glycans, and so basically sugar, the sugar coating of cells is very important for their physiological function. For example, in immune recognition, immune cells have many lectins, so protein that binds to sugar, that sense which type of sugars are on the surface of cells to detect, for example, if it's a virus or bacteria or if it's a self signal. So it's very important in immune recognition, many other physiological phenomena. The difficult thing in this is that to study this interaction, sugar-lectin, is that they're very, very weak. And for many reasons, for this reason, it's been very difficult to study in the field of glycochemistry. But for us, actually, weak is good, because weak means reversible, means we can use it for paint imaging. Actually, we want weak probes. So what we simply did is to take a fluorophore attached to a certain glycan, and this will again float in solution. Then if it finds a lectin on the surface of the cells, will bind with a certain K on. It will surf on the cells together with it during the diffusion, and then will unbind following a certain K off. So the beauty here is that whatever we measure, K on, K off, diffusion, it's all interesting because it's a physiological interaction. So you can tell something with the affinity of a sugar for the lectin, of the diffusion of the lectin, of the expression of this receptor. So whatever we learn from here, it's all useful to understand this biological phenomenon. So the first step for it is just to build a library of sugars, and we could have it done without our collaborator, Jeroen and Sander from Leiden University. They are sugar wizards. They built a whole library of different sugar, for example, mannose in green or sulfomannose in yellow, with different valency, 1, 2, 3, 4, 5, 6. They're all monodispersed precise molecules. They can tune the valency, so the number of sugars. They can change the presentation, a single mannose, a cluster of 2 mannose, a triplet of mannoses. They can change the spacing. So basically, they build a whole library of sugar probes, changing these key features. And the question is how valency, spacing, and type of sugar will influence the binding to the lectin on immune cells. So these are the measurement looks like again. So you see single molecule events of binding of sugars to the surface of the cell. And you see that on the right part of the slides, that depends on the type of sugar, you will see very few events, means very little K on, or a lot of events, means high affinity, high K on. So you can really basically measure something proportional to the K on in the cell. And we can compare. I don't think, if it's one interesting glycochemistry, we can discuss the details of the results, but I think for this audience is not necessary. So feel free to ask about what we found out. Here I want to focus more on the method. Once after binding, we can track the receptors. So we can, again, follow the receptor in time during the movement on the membrane. You can do it for one color or even 2 color tracking. Here we have 2 sugars that binds to 2 different domains. It's mannose in green and sulfomannose in red. And you can see the differential binding and diffusion of the 2 domains in the cell. And we can compare all the different probes if they change in diffusion of the receptor upon binding. And long story short, there's no change in diffusion, means every sugar binds 1-to-1 stoichiometry to the receptor and is not able to, for example, cross-link receptors or induce changes in biochemical behavior. It's a more clean binding. And last, we can measure the K-off, so the time the sugar spent on the protein. And here we measure hundreds of thousands of individual unbinding events fit with the exponential decay, so it's quite good agreement with the theory. And for all of them, again, measure the relative K-off and see which sugar has the highest affinity. What we did at the end, we put all this data together, and on the X and Y axis of this 3D graph, I know 3D graphs looks fancy, but they're often difficult to read. I'll guide you through. On the X and Y axis are basically the K on and K off, basically the density trajectory and the time, residence time of the sugar. So basically the 2 thermodynamic parameter that we can measure with our technique. And the Y-axis is the Z-axis, or the vertical axis, let's call it. There is the functional readout. So which sugar is actually able to internalize in the cells and which one is rejected by the cell. And you clearly see that in this space, there's a space with high K off and low K on that makes the sugar non-functional, but there's a threshold of both parameters. So you need to look at both at the same time to understand this. With low K off and high K on, where it is enough to trigger a functional biological behavior by the cells and trigger, in this case, internalization. This is, by the way, the first step, either in delivery, but also in, for example, in vaccine targeting and processing. So we can really, thanks to this technique, get thermodynamic and kinetic parameters of an physiological interaction in cells. And this brings my conclusion. Very long story short, I think single molecule and supervised technique are beautiful and will allow to understand man-made objects like nanomedicines, molecule by molecule, understand the structure, the interaction with the cells, and we can understand also biology better and which biological feature you want to target with such an object. So hopefully this will guide and help the design of better therapies in the future. But I also think this fundamental understanding is very important to have to give robustness and solidity to this field and get away from just trying and trying molecules. Most important slide, I really want to thank my group for the fantastic work. I acknowledge many people along the way, but this is the whole group. Well, pre-corona photo, we should get a new one actually. And I want to thank also the funding agencies to pay our bills and allow us to buy some few microscopes actually. So thank you, and thank you for your attention. I'll be happy to take questions.
Thank you, Lorenzo, for such a nice presentation. And it's always amazing how you can just see so many things. Not so used to something that is not biologic, but still, I love this spectral super resolution imaging or even the correlative imaging. Actually, my first question will be, how do you align the two images, right? For EM and DNA paint.
Yeah, it's actually one of the trickiest part of the whole workflow. Well, the first tricky part is to find a workflow that's compatible with sample prep is compatible for both methods. This is not obvious. These techniques are not designed to be compatible at all. The alignment is particularly tedious. There's some softwares, but they don't work particularly well. We use stem grids that are labeled, so you can find at least largely the area you're in. And then there's a bit of manual look, try to find the same area. And but the final alignment is done in an easy way with fiducial markers. We use, for example, quantum dots. They're both fluorescent and EM dense. And once you find the same area, you align them, and it's very precise. But finding the same area with two techniques that are different field of view, they're oriented differently to each other, is not trivial. And for cells, it's quite easy, because cells have a peculiar shape. You always find the same cell. Sometimes a funny looking shape and you find it. But particles are all the same. So imagine there's a whole field of view with hundreds of spheres, and they all look the same. So that's really not trivial.
Yeah, I assume it's like looking at constellations in the stars.
It's beautiful, but it's you don't want to be the one aligning. We are trying to make improvements in automated software for detection and alignment. It's semi-automated at this stage.
Okay, good. I'm also very interested in the quantitative measurements that you achieve. How do you account for, I don't know, things like static hindrance, or how do you know that everything that you would like to really count is labeled?
Yeah, that's a very good point. In the case of particles, it's a bit easier because they're very accessible. And you also have more control in what you can do on the labeling. With cells, it is very difficult. We try to compare different antibodies, the different concentration of antibodies to see where we're saturating. But it's one of the reasons, for example, it's very tricky to image, and we don't try not to do it in the basal membrane because it's hardly accessible.
Yeah.
So it's that's really not trivial. But it's, I think it goes all to controls. Long story short, to do a lot of controls, but it's all labeled. And it's again something that goes maybe 60% of the work, and goes in 2 lines of the supplementary to show all the controls, but it's very important.
I can imagine. Yeah, absolutely. So the last question from my side would be, maybe I have missed it, but how do you also label in the live cell paint? Do you also label non-membrane proteins, like from the cytoplasm?
We don't do it. We really have an interest in receptors. Plus, receptor it's easy because, of course, it's accessible from the outside.
Yeah, exactly.
There's been some examples of intracellular labeling, for example, expressing a targeting probe with a GFP or with the allo tag inside cells, and also there see the reversibility to the binding, but it's much more difficult. So in this sense, on one side, we are more interested in receptors, but on the other side, also, it's easier to look at the receptors. So it's a bit of a win-win for us. Intracellular, everything gets much more complicated. Not impossible, but not easy.
Absolutely, yeah. So there are a couple of questions in the audience also. Jan Baumann asked where you obtained the RNA multiplication technique.
Sorry, can you repeat? I didn't hear well.
where did you obtain the RNA multiplication technique? I assume it's the DNA paint, if I understand correctly.
So how do we perform it?
I assume just briefly how do you get this DNA? Where do you get the probes, things like that?
Yeah. So imagine that we can have an antibody that have a certain affinity for a specific target. So we first recombinantly acquire it. Mostly you can purchase, but sometimes you recombinantly express the target itself with the antibody, and then you have to label with DNA. So that's quite a lot of chemical biology there to prepare these probes. This again, labeling is always the key of these techniques. And to be quantitative, you want one DNA per probe. So ideally, you want to express your probe with only one cysteine. They can selectively label one DNA strand, and then you get very quantitative results. Once the DNA, the probe is fished, bound to the particle, and the DNA is there, it's a standard DNA paint type of measurement. That's quite standard, I mean, for the field. And then the sequences are very well known. There's many sequences super validated by us, by other groups. Ralph Human published a lot on it, and so that we just picked the standard DNA paint. But the expression of the probe is very tricky.
Then Aseb Romandar says, what are the benefits for chemistry, industry, and implementation of this technology?
Yeah, good point. We hope a lot. I see two things. The first is to help the discovery. So it's very important once you build anything the characterization is a key step. And now is done a lot with electron microscopy is a key tool. Spectroscopy is done a lot to characterize particles. But I think there's much more that we can be done in terms of characterization. So it's really, if you understand what you produce, you would interpret better the functional results. And once you get into this loop, make a new material, test it, make the next that's supposed to be better. If you understand why it didn't go well, for example, because this is too big, it's too small, it's too little functionality, you'll be faster in making these rounds of optimization. Long term, we hope this will be also a technique used for quality control. If you think about the batch that came out of a whatever company, pharma company, biotech company, it's quite crucial to characterize the batches and the reproducible and robustness. Now it's done a lot by composition, the right things are there, and sometimes a bit of a DLS to check the size and color stability. But I think we'll get more and more strict in time. Also because some particles have a specific functionality bound to having antibodies on the surface or having a certain polarity or having a certain shape. If you don't check that constantly in the batches, you risk to have a non-functional batch. And this happened a lot, to have batches of vaccines or particles that have side effects, more side effects than normal, because they don't comply for all the quality. But that's a more long-term perspective, to be honest. I don't imagine that the FDA will ask tomorrow to check by imaging.
Yeah. Okay. Then Elena Giorgio says, impressive talk. I wonder what is your regular acquisition time with DNA paint using your protocols, and can you quantify clearly with DNA paint, for example, the antibodies, even considering the drift and the intrinsic flexibility of the materials?
Yeah. Good questions. Let's go more technical. We usually have an integration time between 50 and 100 milliseconds for single frames, and give a thumb rule, half an hour acquisition. Of course, you can image longer and you get more events, more statistics, but then of course you lose statistics. So that's it's a trade-off. This I think is a good compromise. We have almost published a paper, I think it was the third round of revision, I think we have done, submitted yesterday evening, that go really in the depth of this, really went through the systematically changing all the technical parameters to maximize precision. That's what you want in counting. So integration time, size of the area you do QPaint, length of measurement. So it's a trade-off, but to give you an idea, we just measure half an hour, 45 minutes. Drift correction, absolutely necessary. And it's always with fiducial markers. The correlation works for certain structure, very, very structured, things like microtubules. But for our sample, drift correction I think is much more effective with fiducial markers. And there was a last question about the flexibility. I would say antibody-antigen interaction is very rigid, so there's not a lot of flexibility there. If your object is, of course, moving, you will lose precision. But I don't think it's a major effect for the large majority of the samples. I mean, still, even DNA paint, you have 10, 15, 20 nanometer precision. A 20 nanometer vibration is actually quite a lot for a molecule, so I wouldn't say it's significant. If you do mean flux or things a bit even more sophisticated, then maybe start to play. But we don't worry about it so far.
Okay, yeah, good. She also says, I also found interesting the difficulties with the two-color storm experiments. Can you explain the main problems and what are the best fluorophores in your opinion for storm?
Yeah, that's a very good question. I would say we're moving out a lot to paint also because multicolor storm we find it quite difficult. It's not impossible. We saw with our reports, we do it as well. So, but especially if you want to be quantitative, there is the Alexa 647, it is in the heaven of dyes. Dyes that are like Alexa 647, so very similar. So the CF 647, basically whatever 647 generally works quite well. Work like a charm. Any buffer you try start to blink, perfect. All the rest is on a completely different league. So we optimize a buffer, we try Oxia, different locks. You can get a second color okay-ish, but it's always, if you want to quantitate, give numbers, we don't trust it. So for structural imaging, I think it's still okay. But if you really want to count, it's really difficult in our hands. Yeah. I know other people publish, but again, I didn't see counting with that.
And it's a difficult thing, yeah.
It's very difficult. Also, you have to trust it, and it depends also on the density. If it's a not very dense object, the blinking is less crucial, so you can still count okay. But for dense object, it's really difficult. So that's why we chose this approach, having two red dyes and separate them by the spectral imaging. You can also do spectral demixing is another approach, but we already have the setup built. We didn't show it today. We did up to 4 color imaging of red dyes, like 633, 645, 680, 700, and it still works.
Nice. Then Satakshi S says, how would you quantitatively evaluate the surface interaction of the nanoparticle and receptor? Any specific characterizations or theoretical tools?
Yeah, there's a lot to do there. I think you mentioned theoretical tools. We work a lot with theoreticians to do simulations. There's Dan Frankel is the absolute god of them. He published a lot of methods, theoretical methods to estimate and simulate interaction of multivariant objects to a multivariant surface. We try to actually see if our data match the simulation of Dan Frankel. You can think about that if to match a theoretical model, you need data, quantitative data on. So counting receptor and counting ligands is exactly what you need to inject into a model to predict which particle is good or not. So I think that's a bit our strategy. If you have a library of particles and a library of cells and see who match to who, who's the right targeting to who. If you count receptors and count ligands and particles with the DNA paint, then you can inject into a theoretical model. They can predict who is the most functional particle and then select from the library. It's still work in progress. Methods now are there, I think, in the last years, and now we are getting more and more into having libraries of particle and go to functionality. But if you didn't see them, look at the work of Dan Frankel. The theory is astonishing.
Great. Then Umar bin Adnan says, can we use glycopaint to label receptors of neurons?
In principle, I say nothing against. I was actually invited some months ago to speak in a neuroscience conference for the first time. I'm a chemist by training, so as far as you can get from a background. The beauty of the glycopaint or lifestyle paint is that you don't need any labeling, because it's basically taking a dye with a sugar, put in the solution, and measure right away. So that, especially for cells that are sensitive like neurons, they're difficult to transfect, difficult to handle, it could be an advantage. So once I see no reason why it should not be used to measure sugar receptors on neurons. Is actually The methods, it's difficult to develop the probe. Once the probe is rather easy. Even the journal, when we published, asked, can you extend a bit the methods, because it's a very technical paper, can you write more? But then it was actually all about it. Is add 1 nanomolar sugar label in the solution and press start to the microscope. So it should fit particularly weak, difficult cells like neurons, primaries.
I would like to test that with the background in neurons and labeling receptors. It would be very interesting to be more quantitative on that side.
We can share some sugars. The beauty is that we need very little amounts. So as is paint is nanomolar, picomolar concentration in the medium. You have to say the synthesis of these sugars is not trivial. They're really good sugar chemists. The groups that do only that, they're very difficult to make, but we need very little. They were happy about it.
Good. And then Zupuri says, could you please share a few key components required to initiate the fabrication process of novel nanomaterials?
Oh, to publish novel nanomaterials, they should talk to editor of journals then. It's I think it really depends what's the novelty, if it's synthetic or characterization or application. I mean, there's a whole world of nanomedicine, nanotechnology journals, but you really focus what's the novelty, and it depends on that. There's a key standards of reporting synthetic procedure, reporting characterization. You can reach by email with maybe more details. But you should talk to editors. I publish, but I don't decide who publishes.
Yeah. Absolutely. Okay, so I think this is everything that we have. Finally, thank you, Dr. Albertazzi, for joining us today. Thank you, the audience who has watched us and followed with questions. If you have any information regarding our technology or what Lorenzo has had, you can reach us through our website at oni.bio or Twitter or LinkedIn accounts. We'll be happy to see you in the next webinars. And thank you, everyone, for joining us. Have a good day, morning, afternoon.
Thank you for listening. If you have further questions, you can reach me by email.
Absolutely. Thank you, everyone. Bye.